I want to be honest with you all, this is going to get difficult to understand from now on. Now we're going to talk about the biomolecules that make the magic happen on the field in every living being, from a bacteria to your ankle, from a squid to an oak tree. Whenever your body is fighting against an infection thanks to antibodies, or when you feel sad or happy because of neurohormones, or even when you breathe and oxygen needs to go to all of your cells, it's always a protein that makes it happen. They're so important that it's even written in their name. Pro comes from Greek, and it means primary, since they make everything you need to, well, to be alive. Now, how is that possible? How can a single class of biomolecules do all these very different kinds of tasks? First of all, we need to know what they're made of. The answer is amino acids. They are a family of chemical compounds which share a similar aspect. They are made of a carbon atom that makes a bond with an amino group NH2, a carboxyl group COOH, which is a carboxylic acid. By the way, that's why they're called amino acids. I didn't come up with the name, okay? A hydrogen atom and the side chain R, different for each amino acid. What is this R chain? It is a chain made of different molecules depending on which amino acid we're talking about, and this is what makes every one of them different from one another. The chain gives different chemical properties to the specific amino acid. Some are polar, some are not, some are linear, some are cyclic, some contain sulfur, some contain benzene. Thanks to the differences in the side chain, we can distinguish the different amino acids. Now, even though there are technically hundreds of different amino acids, only 20 of them are what we call natural, and only these 20 make the proteins. Problem is, our human body doesn't know how to build 9 out of these 20 amino acids, so we need to acquire them thanks to the diet. They are what we call essential amino acids. Now, all of these different features are what they need to make the magic happen. In this context, by magic, I mean an incredibly complex and potentially infinite number of completely different molecules. How do they do it? Well, first of all, they take a bit of inspiration from the carbohydrates. Remember how they could make long chains thanks to the glycosidic bond? Amino acids do pretty much the same thing, making long chains thanks to the peptidic bond, a covalent bond between the amino group from an amino acid and the carboxyl group of the next amino acid. How do they actually choose what amino acids want together, in which order, and how they make the bonds? They are all incredibly complex topics that require you to know something about DNA, RNA, and ribosomes, but we'll talk about it in some future videos. Fun fact, remember that I told you about how molecules with the same formula may be different from one another, forming different isomers? Right, well, amino acids come in two different flavors, the L and D configurations. Now, for some reason that we're still trying to understand, the greatest majority of all the proteins in every living being is made from L amino acids. Is this information useful in some way? Spoiler alert, nope. Anyway, you now know that proteins are made from chains of amino acid, except they are not, because it's way more complicated than that. To fully understand what is going on here, we need to talk about the 4 different structures of a protein. First of all, we need to clear something out. When you link together a bunch of amino acids, you don't find yourself in the presence of a protein, but only of what we call a polypeptide. Well, a protein is one or even more polypeptides linked together that has also reached a 3-dimensional biologically active stable structure called native conformation. We can make the difference between monomeric or multimeric protein whether it's made of one or more polypeptide, but in either case it has to reach the native conformation to be properly called a protein. Now, since they are made of amino acids, we can say that every protein has an intrinsic directionality, with an aminic group to one end, the N-terminal end, and a carboxyl group to the other end, the C-terminal end. Even though only the 20 natural amino acids build the proteins, they can combine in pretty much infinite ways, and so the quantity of proteins is ginormous or something like that. The mere differences between amino acids aren't enough to explain all of this though, because, as I said in the last video, proteins are way more complex than that. We need to talk about the 4 different structures of a protein. What we call the primary structure is simply the sequence of the different amino acids that make the backbone of a polypeptide. This is only the beginning though. Things start to get interesting with the secondary structure, more or less long regions of the chain folded repetitively and regularly, forming the alpha helixes and beta sheets. These regions are linked together thanks to hydrogen bonds that are formed between the atoms of the peptide skeleton. Note that this does not involve the lateral chain R, and since there are a lot of these structures inside a protein, even though they are only made of hydrogen bonds, they end up making it much more stable. All the other parts of a protein that are not part of neither alpha helices nor beta sheets are called randomly wound regions. Moving on, we find the tertiary structure, which is formed through bending and folding of the whole molecule and is the consequence of interactions between the R groups of the different amino acids. 4 different kinds of bond can help to make this structure: the hydrophobic co-hydrophilic interactions, the ionic bonds that form salt bridges, the hydrogen bonds, and disulfide bridges. Last but not least, we found the quaternary structure, which is composed of the mutual interactions, and by that I mean the same kind of bond that occurs in the tertiary structure, of the individual monomeric subunits that form a multimeric protein. This means that only multimeric proteins present a quaternary structure. The classic example of a multimeric protein is hemoglobin, a protein made of 4 different polypeptides that are linked together. This is the most abundant protein inside red blood cells, and is the one responsible for the transport of oxygen around the body, that forms a bond with particular region inside the hemoglobin called in group. With all of this in mind, you can start to understand why proteins are so complex. Aside from being really big molecules, they also form these 4 different structures and behave accordingly in completely different ways. The different interactions of the structures of a protein determine the native conformation, which allows us to categorize proteins in 2 big families: fibrous proteins, which are very long with a big secondary structure, and the last sentence was better in my mind for some reasons. They usually play a structural role outside cells. Some examples are keratin, collagen or elastin, and globular proteins. They have a compact shape and represent the great majority of the proteins inside a cell. That said, I still covered only a fraction of what proteins actually are, but we'll understand the missing parts in future videos, where we'll cover how amino acids know in which order they're linked together to form a protein, and the whole process of the maturation of a protein. As for the moment being, this is enough to move on with the next topic, which right now consists of the most famous molecules around, the nucleic acids, RNA and DNA.